Welcome to Deep Dive Lab, where we break down and dissect cutting-edge scientific research, from advanced biomedical technologies to breakthroughs in AI, material science, chemistry, and physics, all told as compelling, easy-to-follow stories. And today we are diving into a biological mechanism that is really common, but we are looking at it in a completely unexpected way.
Yeah, it's a really fresh perspective on something we thought we basically already had figured out.
Right. So to start, I want you to imagine managing your cholesterol, managing the door at a wildly overbooked Hollywood nightclub. It is packed, the music is deafening, and the VIP section is just overflowing with people who probably shouldn't be there.
That is a great visual. And to keep things from just, spiraling into total chaos, you really have to rely on your bouncers.
Exactly. In your cardiovascular system, these bouncers are your low-density lipoprotein receptors, or LDLRs for short. And their sole biological purpose is to stand at the membranes of your liver cells, grab the rowdy circulating bad cholesterol, which is LDLC, and just physically yank it out of the bloodstream.
It's a beautifully brutal clearance system, really. I mean, those LDLR bouncers are the primary defense mechanism keeping the walls of your arteries free from plaque accumulation.
Right, so the higher the receptor density on the surface of those liver cells, the cleaner the bloodstream.
Precisely. We rely completely on that density to stave off atherosclerosis.
So you want a massive security team working the door at all times. But here's a strange question. Why does eating a high cholesterol diet cause your body to actively destroy its own bouncers?
I know, it sounds like a massive biological paradox. You would naturally assume that, well, when the bloodstream is flooded with dietary LDL, the liver would upregulate its defense, right? Like hire more bouncers to clear the influx.
Yeah, that would make sense.
But instead, the liver initiates this targeted self-destruct sequence. It actively pulls the receptors off the cellular membrane and intentionally eliminates them.
Okay, let's unpack this, because if I'm tracking the logic here, the body is responding to a systemic metabolic crisis by basically throwing away the only tool it has to fix it.
That is exactly what is happening, yeah.
But before we get into the mechanics of this self-sabotage, we need to contextualize the current medical landscape. Because I imagine you, the listener, are probably thinking about the standard of care right now. Don't we already have drugs like statins or PCSK9 inhibitors for this?
Right. Millions of people take those every day.
Yeah. So why is this disappearing bouncer phenomenon still considered a critical mystery if we already have the pharmacology to handle high cholesterol?
That assumption is incredibly common, even within broader medical discussions. Statins and PCSK9 inhibitors are, without a doubt, absolute titans in cardiovascular medicine.
They do work.
They do. They drastically lower LDL. But they are far from being perfect magic bullets. I mean, they come with significant clinical baggage that limits their utility for a really large subset of the patient population.
We're talking about the severe side effect profiles, right? Like the collateral damage of those blunt instrument mechanisms.
Exactly. Hepatotoxicity and myopathy are the classic hurdles with statin intolerance. Patients experience severe muscle pain or, elevated liver enzymes that literally force them to discontinue the therapy.
Oh, wow.
And furthermore, prolonged high-dose statin therapy has a well-documented association with an increased risk of new-onset diabetes.
Wait, really? So you fix one problem but create another?
Yeah, you are essentially shifting the patient's risk profile from one metabolic vulnerability to another.
And the newer injectable drugs, the PCSK9 inhibitors, those bypass some of the muscle and liver issues, but they introduce a whole different set of complications, don't they?
They do. I mean, they preserve the LDLR bouncers highly effectively, but they require chronic subcutaneous injections, which presents a major barrier to long-term patient adherence. Not everyone wants to inject themselves constantly.
Plus, the economic cost is staggering, right?
Oh, it's massive. And beyond that, broader post-marketing surveillance over the last few years has flagged various adverse events, including unexpected metabolic disturbances and, in some cohorts, neurological and cardiac issues. So, yes, we have treatments, but we desperately need better, more targeted oral pathways.
That brings us to the core of this biological black box. Because we have known for a long time that dietary cholesterol lowers LDLR expression.
Right, we knew the levels dropped.
But the confusion was always about the post-translational pathways.
Right.
Like, we understood that the body stops making as many new receptors, and we understood the role of circulating regulators like PCSK9.
Yes, the well-known pathways.
But even factoring all that in, there was this invisible, aggressive degradation process happening. The receptors were being systematically murdered after they were billed, and nobody could see the executioner.
It was a massive gap in our understanding of cellular trafficking. The liver creates the receptor, deploys it to the membrane, and then under high cholesterol conditions, it just vanishes, entirely independent of those known PCSK9 pathways. The mechanism was basically a ghost.
So how did scientists figure this out? This is where the story gets interesting.
Well, we are looking at a landmark study published just this week on June 24th, 2026 in the journal Nature. The research was led by Dr. Zhu Feng, Dr. Alan R. Saltiel, and a team of colleagues from the University of California, San Diego.
UCSD, got it.
Right. And they essentially decided to map the entire life cycle of the LDLR under severe dietary stress.
And to do that effectively, they utilize a highly controlled in vivo model. They placed mice on a high cholesterol diet, or HCD, and tracked them over a prolonged timeline, up to 20 weeks, actually.
Which is huge. I mean, in the lifespan of a laboratory mouse, 20 weeks is a massive duration.
Right. It's designed to mimic years of chronic compounding dietary stress in a human being, like eating fast food for a decade.
Exactly. And the temporal aspect of their experimental design is what really unlocked the mystery. They didn't just look at the endpoint, you know? They tracked the degradation at 4 weeks, 12 weeks, and 20 weeks.
And what did they see at the start?
Well, at the 4-week mark on the high cholesterol diet, the LDLR levels on the cellular membranes were completely stable.
Oh, really?
Yeah. The bouncers were maintaining the perimeter perfectly. The biological system was successfully compensating for the influx of fat.
Okay, so the club is packed, the music is blaring, but the staff is holding the line. The system hasn't broken yet.
Precisely. But sometime between week 4 and week 12, the entire regulatory structure just collapses.
Wow.
Yeah. By week 12, and profoundly by week 20, the LDLR protein levels plummeted. The bouncers disappeared from the membrane, and the circulating LDL cholesterol in the blood skyrocketed.
So the system just gave up.
It did, and Dr. Feng and Dr. Saltiel zeroed in on that specific window to figure out what molecular switch got flipped by that chronic stress.
And they mapped the entire sequence. And the primary culprit turned out to be a family of molecular switches known as RAL GTPases, specifically the proteins RalA and RalB.
Yeah. Think of these RAL proteins as middle managers within the cell's intricate logistics network. Their standard biological function is to direct intracellular traffic.
Right, keeping things moving.
Exactly. They toggle between an inactive GDP-bound state and an active GDP-bound state.
But under the crushing weight of a chronic high cholesterol diet, those middle managers essentially get bribed into working against the company.
That's a great way to put it, because the initial signal actually comes from higher up the chain. Chronic cholesterol exposure hyperactivates a very famous upstream signaling protein called RAS.
RAS, okay.
You can view RAS as a panicked senior executive. When the liver is constantly bombarded with lipids week after week, the hyperactive RAS executive reaches down and permanently locks those RAL switches into their active state.
So they're just stuck in the on position.
Exactly.
Now you have these RAL middle managers, wide awake, highly active, and receiving constant distress signals from RAS. And their response is to engage an endocytic complex, specifically recruiting two proteins named RalBP1 and REPS1.
Right. Those two are key.
This newly formed complex acts like a coordinated kidnapping crew. They physically latch onto the LDLR bouncers at the cell surface and just drag them inside the cell.
Which is wild, but the nuance here is critical because internalizing receptors isn't inherently malicious, right?
It's normal sometimes.
Yeah, under standard physiological conditions, an LDLR grabs an LDL particle, pulls it inside the cell, releases the lipid, and then a dedicated adapter protein called SNX17 grabs the receptor by its tail and recycles it right back to the cell membrane.
Ah, okay, so it's a highly efficient, sustainable loop. The bouncer escorts a patron out, then walks right back to the front door.
Exactly. But the RalBP1 and REPS1 kidnapping crew actively prevents that return trip.
They hijack it.
They completely hijack the sorting endosome. The active RAL complex physically outcompetes or bypasses the SNX17 recycling mechanism entirely.
Oh, wow. So where do they take it?
Instead of routing the receptor back to the surface, the RAL crew drags the LDLR deep into the cell's lysosomes.
And the lysosome is basically the biological incinerator. It is an acidic, enzyme-rich organelle explicitly designed to break down cellular waste and macromolecules.
It is, and the ultimate executioner waiting inside that lysosome is a specific protease known as Cathepsin A, or CTSA.
The shredder.
The shredder. Once the kidnapped receptor is delivered into that acidic environment, Cathepsin A shreds the LDLR protein down to its constituent amino acids. the bouncer is gone forever.
Man, let me synthesize the scale of this cellular betrayal because it's crazy. It's as if a city is dealing with a catastrophic overflow of garbage.
Hmm?
And instead of sending the garbage trucks out to clear the streets, a panic executive RAS orders a rogue manager RalA to divert every single garbage truck into a fiery incinerator where they are melted down by Cathepsin A.
Yes, that is the exact pathway.
And then the streets just fill up with trash. But I have to push back on the evolutionary logic here. Why on earth would the body evolve to do this? It seems entirely counterproductive.
Yeah.
You don't build a mechanism that actively sabotages cholesterol clearance during a period of lipid surplus.
What's fascinating here is the concept of evolutionary mismatch. The human body, and the mammalian system in general, did not evolve to handle a 20-year, unrelenting influx of saturated fats and double cheeseburgers.
Right, we used to have to hunt and forage.
Exactly. In a metabolically healthy system, transient, short-term activation of RalA is perfectly normal. It helps remodel the membrane or adjust receptor density based on acute temporary needs.
So it's supposed to be a brief adjustment.
Yes, but chronic activation entirely reprograms the signaling architecture. The constant metabolic stress essentially short-circuits the pathway. A system designed to make microadjustments gets locked in the on position, turning a basic trafficking function into a runaway destructive loop.
So it is an acute regulatory system collapsing under the weight of a chronic modern environment. That makes sense from a pathological standpoint. But, let's look at the leap from a highly controlled animal model to human cardiology.
Sure, that's the big question.
Because a mouse eating a synthetic high-fat diet for 20 weeks is an extreme artificial stressor. And a mouse only lives for about 2 years anyway. How do we know this specific kidnapping and incinerator pathway isn't just an evolutionary quirk unique to rodent biology?
Well, the UCSD team anticipated that exact skepticism, which is why the second half of this Nature paper is completely dedicated to human genetic validation using genome-wide association studies, or GWAS.
Oh, so they looked at real human data.
They did. They cross-referenced their molecular findings with the genetic data of hundreds of thousands of human beings.
They were looking for structural variations in this newly discovered pathway, right?
Yeah.
To see if it dictates real-world cholesterol levels in humans.
Exactly. They analyzed the genetic loci for the exact proteins involved in this cascade: the KRAS gene, the REPS1 gene, and the CTSA gene.
And what was the hypothesis there?
Well, if this pathway is active in humans, individuals with slight structural variations or polymorphisms in these genes should present with altered lipid profiles. And the data overwhelmingly confirmed it. Variations in these specific genes are directly and strongly linked to altered total cholesterol and LDL levels in the human population.
So they mapped the molecular addresses, essentially, and they found a specific genetic variant, RS75667995, located in the KRAS gene. Human beings carrying that specific sequence variation demonstrate significantly different serum cholesterol baselines.
Yes. And they found another highly impactful variant, RS729747222, situated in the REPS1 gene. Remember, that's the protein responsible for physically tethering the endocytic kidnapping crew.
Right, the kidnappers themselves.
So if a human has a mutation that makes that REPS1 protein slightly sluggish, or conversely hyperactive, it drastically dictates their circulating LDL-C levels. This genetic evidence confirms that the incinerator pathway is actively governing human cardiovascular risk.
Wow. So if we have this complete schematic, the executives, the rogue managers, the kidnapping crew, and the incinerator, that shifts this from a basic biology problem to an engineering problem. Can we build a pharmacological roadblock?
Yes.
Because the study didn't just map the problem, right? They tested actual molecules to interrupt the sequence.
They attacked the pathway from two distinct pharmacological angles. The first approach utilized a small molecule called RBC8, which functions as a direct RAL inhibitor.
Now, targeting GTPases like RAS or RalB has historically been incredibly difficult. I mean, they were often considered undruggable because their surfaces are so smooth and they have a massive binding affinity for GDP. How does RBC8 manage to shut down the rogue manager?
It's really clever. RBC8 manages to bind to an allosteric pocket on the RAL protein, effectively stabilizing it in its inactive GDP-bound state.
It just freezes it.
Basically, yeah, it strips the manager of its ability to communicate. Because RAL can no longer activate, it cannot recruit the REPS1 kidnapping crew. And when they administered RBC8 to the mice suffering from the chronic high cholesterol diet, it restored the LDLR levels on the liver membranes and triggered a rapid drop in circulating bad cholesterol.
So you take away the manager's radio, and the garbage trucks never get diverted. That is amazing.
Yeah.
But they also tested a second intervention, one that ignores the kidnapping entirely and focuses directly on the incinerator.
Right. The second drug is a compound called AS-R164653. This is a highly specific CTSA inhibitor. It targets Cathepsin A.
My immediate concern with inhibiting a lysosomal protease, though, is the collateral damage. The lysosome is the cell's waste management system for everything. If you shut down Cathepsin A, don't you risk wrecking the cell's ability to clear out other toxic protein?
It is a totally valid concern, but Cathepsin A is remarkably specific in its proteolytic targets within this context. SARO-164653 inhibits the shredding action of Cathepsin A without globally disabling the lysosome's acidic breakdown of other necessary macromolecules.
Oh, that's a relief.
Yeah, so when the bouncer gets dragged into the lysosome, the CTSA inhibitor acts like a protective shield. The receptor survives the acidic environment, escapes degradation, and is eventually routed back to the cell surface. Treating the in vivo models with SAR-164653 comprehensively rescued the LDLR proteins and massively improved cholesterol clearance.
So what does this all mean? We have a stunningly detailed map of a previously invisible disease pathway, and we have 2 targeted molecules that successfully block it in animal models. But translating a Nature paper into a pharmacy prescription usually takes, 15 years of grueling clinical trials. How close are we to actually deploying this against human cardiovascular disease?
The timeline here is honestly the most exciting revelation of the entire discussion. The CTSA inhibitor, SAR-164653, is not just an experimental lab chemical. It has already successfully passed Phase I clinical safety trials in healthy human subjects.
Wait, it is already navigating the human regulatory pipeline.
It is. It was originally being investigated for other cardiac remodeling and heart failure indications, which means the fundamental pharmacokinetics, tolerability, and safety profiles in humans are already established.
That is incredible.
We are remarkably close to introducing a brand new class of oral cholesterol medications based on this exact mechanism.
Think about the clinical paradigm shift that enables. Instead of forcing a patient to endure high-dose statins and risking severe hepatotoxicity or muscle degradation, a physician could theoretically prescribe a low-dose statin combined with a CTSA inhibitor. You gently suppress cholesterol synthesis with the statin, and you forcefully protect the existing LDLR bouncers with the CTSA inhibitor.
Exactly. You achieve profound lipid clearance synergistically. It would eliminate the need for expensive, burdensome subcutaneous PCSK9 injections while sidestepping the severe metabolic collateral damage of heavy statin use. It is precision pharmacology replacing blunt force.
We are fundamentally changing the strategy. We are no longer just trying to empty the nightclub. We are finally ensuring the security team stays on the payroll.
I love that analogy.
Let's synthesize the crucial data points from this UCSD research for everyone listening. The complexity really boils down to 3 primary takeaways. First, chronic dietary cholesterol doesn't just passively accumulate in the bloodstream. It actively triggers a hostile, self-destructive cellular signaling cascade driven by the RAS and RalB proteins.
Second, this hyperactive pathway physically recruits endocytic proteins to force your liver's critical cholesterol-clearing receptors into cellular lysosomes, where they are specifically shredded by the Cathepsin A enzyme.
And third, by mapping the genetics of this execution sequence, scientists have demonstrated that inhibiting Cathepsin A with small molecule drugs offers a highly viable, rapidly approaching oral alternative to statins and PCSK9 inhibitors.
If we connect this to the bigger picture, it is a profound testament to the delicacy of our cellular architecture. These signaling pathways evolved over millions of years to manage caloric scarcity and make microscopic adjustments to our metabolism.
Right, they are ancient systems.
And when we inundate those ancient sensitive systems with the chronic stress of modern high cholesterol diets, the biology doesn't just struggle to keep up, it actively misinterprets the signals and weaponizes our own cellular machinery against us.
It entirely recontextualizes how we view a poor diet. You aren't just consuming excess lipids, you are flooding your system with a biochemical distress signal that literally reprograms your cellular managers to destroy your own defenses.
That is exactly the reality of it.
But the immense silver lining here is our capacity to decipher that reprogramming. We opened the black box, we found the rogue managers, and now we are engineering the pharmacological rescue operation. If this discovery is just the beginning, what might scientists uncover next?
The cellular landscape is vast, and we're really only just beginning to map the incredible mechanisms hiding in plain sight.
Thank you for joining us in the Deep Dive Lab, and be sure to follow and subscribe for more science stories.